One of the actions of insulin is to promote glucose uptake by certain cell types. So I guess I should actually preface by saying that most cells don't have regulated uptake of glucose. Glucose will just enter the cells by uh, facilitated diffusion. But in certain cell types, such as adipose and resting muscle cells, um, the uptake of glucose is insulin dependent. Okay. And this slide right here shows us how that happens, the mechanism by which these cells are going to take up glucose. Okay. So looking at the left panel here, we have our cell, our adipose or muscle cell right here. Okay. Um, if insulin is absent, glucose can't come into the cell because there aren't transporters for the glucose to enter these cells. Okay. When we have insulin present, what happens is insulin is going to encourage or stimulate the fusion of these secretory vesicles to the plasma membrane. These secretory vesicles have in them a transporter called GLUT4, which is capable of transporting glucose across the membrane, right? So insulin is going to bind to a receptor that is on that cell type, lead to a signal transduction cascade, which is going to lead to the insertion of these GLUT4 transporters into the plasma membrane of these cell types, the adipose and muscle cell types. And then with those GLUT4 transporters present, we are then able to transport glucose across the plasma membrane. Now I just wanna quickly chime in and say, kind of comment on why these particular cell types are insulin dependent. So you might remember that these are the cell types, these two cell types, along with the liver cells, are the ones where we are going to be storing our energy in the form of glycogen or fat. Okay. Um, and if we are in the fasted state here, okay, um, we're not going to want to do that. We don't want to store away our glucose. So we are going to want to do this. Okay. However, if we are in the fed state, then we can secrete insulin, which is then going to tell these cells, okay, it's okay to take up glucose and start building that glucose up into larger storage molecules. Okay. All right. So I think I basically, in this explanation, just basically answered this question, but a quick concept check here. Which of these is going to represent the fasted state? Which is going to represent the fed state? I pretty much already gave you the answer, but think about it for a second. Okay, so the left is going to be the fasted state, right? So if we don't have a lot of nutrients available, we don't want to be taking glucose up into these cell types to store them away as storage molecules. We want that glucose to stay in the bloodstream where it can be prioritized for delivery to other cell types, particularly the brain, okay? On the right side, that's going to be the fed state. If we have a lot of glucose around, we're going to be in a position where we can afford to take up glucose into these cells and store that glucose in our longer molecules, larger molecules. Okay, finally, I just kind of want to mention that this is for adipose and muscle cells. We also have glucose uptake by liver cells that is insulin dependent, but that uses a different mechanism that I'm not going to talk about. Okay, so a couple slides ago, I showed you that insulin is going to favor anabolism. But I want to take a moment right now to address this question of, well, what does that mean? What does it mean to favor anabolism? How do you do that mechanistically? How do you promote anabolism? And this slide basically illustrates how we do that. Okay, so All of these metabolic pathways that I have been describing consist of individual chemical reactions that are catalyzed by enzymes. Okay, so you'll have one enzyme catalyzing the forward reaction and another enzyme catalyzing the backward reaction. If you are interested in promoting a particular pathway or the formation of a particular product, such as glycogen, what you can do is you can activate or upregulate the enzymes that are going to be catalyzing that reaction. Okay, so that's what's shown here. Likewise, if you want to favor the formation of the precursor molecule, right, if you want to go backwards, then you can upregulate either by upregulating the expression or modifying that enzyme in some way to make it more active. You can upregulate the activity of the enzyme that is going to be catalyzing the backwards reaction. So in this case, the breakdown of glycogen to glucose. Okay. So in the presence of insulin, we are going to want to make more glycogen. We are also going to want to stimulate pathways that are going to favor anabolic processes: glucose oxidation, glycogen synthesis, fat synthesis, protein synthesis. Okay, so that basically covers our actions of insulin. I just want to close with this slide, which summarizes the actions of insulin. I want to remind you that insulin is made by the beta cells of the pancreas. Okay, the beta cells of the pancreas are going to be stimulated to secrete insulin by a few different stimuli. We have these stimuli from our digestive tract. So you may or may not remember this from the digestive unit, but when our stomach distends, okay, so when it grows from the entry of food, that is going to activate the nervous system, which then is going to increase parasympathetic activity, and that is going to stimulate the beta cells. Also, the presence of nutrients in the GI tract is going to lead to hormonal secretions, which are then going to stimulate the beta cells to secrete insulin. And then finally, what we talked about here in this set of slides, we have our increased nutrients, amino acids, and glucose in the bloodstream, which are directly going to stimulate insulin secretion from the beta cells of the pancreas. In response to insulin, we are going to increase glucose transport into our insulin sensitive cells, and we are going to promote anabolism. And that then is going to lead to a fall in plasma glucose.